is the microbiological diagnosis last part, that is the microbiological diagnosis, which is the last test of the lab diagnosis. Okay, for microbiological diagnosis, here the serology plays the major role in diagnosis. Okay, first the detection of antibody, as the IgM demonstration in serum provide the early diagnosis, and the IgM appears 5 days after the onset of the symptom and persists for 1 to 3 months. The ELISA test is used to detect the IgM, and for rapid diagnosis of IgM, there is a strip of immunochromatographic test is done. Okay, next the IgG appear later the IgM, for which ELISA is not used to detect the IgG. Okay, and here you have aroused question in your mind, why the IgM is first appear and later the IgG. Wait, I am answering that. When you have contaminate or contact with some new antigen, new foreign antigen, and you have the initial stage of infection, the immune system produces IgM antibody in the initial stage, and later at IgG. Okay, because the positive and the positive IgM test indicates that you may have been infected, and that your immune system has started responding to the virus. There are the natural antibody. The first antibody produced in a human response are always the IgM, because it can express without isotype switching, and IgM produced before B cells have undergone somatic hypermutation and so tend to be low affinity. And here another question arose in your mind, that is isotyping. What do you mean by isotype switching? And the isotype switching is as the is the biomechanism that always B cell production of immunoglobulin from one type to other, such as IgM to IgG. It is the biological mechanism in which it always changes the B cells production from one immunoglobulin type to other. As our immune system produces IgM in the initial stage of infection, that's why due as it can express without the isotype switching, but IgG needs isotype switching. That's why the IgM takes the first appearance when you have in the initial stage, and later by using the isotype switching, IgG appears in the latter stage. Okay, and this IgM is specialized to activate the complement efficiently upon binding antigen, and as this IgG high affinity and found in blood and in extracellular fluid, they neutralize toxin, viruses, and bacteria, and opsonize them for phagocytosis and activate the complement system. So here the IgG have more and high affinity, okay, than the IgM. Here the next microbiological diagnosis, that is the detection of NS1 antigen. Here the immunochromatographic test is used for detecting the NS1 antigen, and its rapid test just takes 15 minutes to detect the antigen on the first day fever before antigen appears. This is the best opportunity to find the NS1 antigen, which is the best test. Okay, and next the isolation of virus. Here, virus isolation done can be done by inoculating clinical specimen in mosquitoes and mosquito cell lines, which is the cell lines is of AP61 cell line, and the suckling mice. Okay, and its identification is done by fluorescent antibody test. Here, the NS1 antigen can be identified by the fluorescent antibody test, and its it's sensitive test also done by taking live mosquito inoculation. Okay, and the next one is the RT-PCR. What do you mean by RT-PCR? Its full form is reverse transcriptase polymerase chain reaction, by which RNA can be detected by in clinical specimen by using this RT-PCR. Viral genome can also be detected. And the last one is the prophylaxis. Okay, as no vaccine is available, so you have to eliminate the mosquitoes and use the control measures, the elimination of mosquito using mosquito net, mosquito repellent cream, and now the live attenuated vaccines are on you and vaccines containing all dengue serotypes under clinical trials. Now you just remember, you just memorize that all four serotypes that are den 1, den 2, den 3 and den 4. These are the live attenuated, these are all the four serotypes of the dengue virus, which are using for trials for preparing the live attenuated vaccines. That's all. Thank you.